Okay, everyone, it's 9 o'clock Pacific time. It's all kinds of hours where you are. I want to thank you for joining us. Monkol, I want to make sure you're there and ready to begin after I introduce everything.
Yep, I'm here.
Okay, perfect. I just want to thank everyone for joining us. NDF is so happy and proud to be able to present opportunities like this for people, stakeholders of GNE myopathy all over the world to join us and to learn from the science and all of the things that we're doing on behalf of the GNE myopathy community. I'm really excited to tell you that we had a great response to this webinar, and we've got people joining us, or at least registered, from countries like Israel, Italy, India, Turkey, Pakistan, United Kingdom, Canada. I'm sure there are others, and then many of the states in the United States. So we are definitely a diverse group, and I know that we're all interested to hear from Monkol, so I won't waste a whole lot of your time with the administrative matters. I just want to make sure that everyone understands to please keep your microphones muted so that Monkol can make his presentation without background noise coming in from everybody else. Feel free to type in the chat any questions that you'd like to address with Monkol. I will try to get them answered, if not during his presentation, then certainly afterwards. And hopefully everything that everyone wants to know, he'll be able to address, at least with regard to the topic that he's addressing today. I want to introduce Monkol Lek. Some of you have met him at some of our events. We're so honored and pleased to have him in our midst and as a team member of NDF. He works closely with all of our scientists and all of our staff as our scientific director. Monkol received his undergraduate degree in computer engineering from the University of New South Wales and then worked for IBM. He returned to UNSW and completed undergraduate degree in physiology and engineering, bioinformatics, and received the University Medal in 2007. He completed his PhD at the University of Sydney in 2012 and did his postdoctoral training in Daniel MacArthur's lab based in Massachusetts General Hospital, Harvard Medical School, and the Broad Institute. So quite an academic pedigree that our Dr. Lek has. He has a strong passion for rare muscle disease research as a patient with limb-girdle muscular dystrophy, and he currently is an assistant professor and research group leader at the Yale School of Medicine's Department of Genetics. Did I miss anything? Well, I'm sure I did, Monkol, but obviously all very impressive, and we're all just so happy that you have taken GNE myopathy in among your interests and the work that you've done for us and for our patients worldwide. So with that, I will leave it to you and your presentation, and I will be standing by to help moderate any questions that come in during your presentation.
Cool. Thank you very much, Nancy, for the very, very kind introduction, a very long introduction too. I should give you a shortened version of that. But just to summarize, a very short version of that, I spent way too long in university learning stuff. But I'm very passionate about neuromuscular disease research. And for those that haven't met me, one of the reasons why I'm passionate about neuromuscular disease research is I have a form of neuromuscular disease myself, a limb-girdle muscular dystrophy, and that's why I'm so passionate about, advancing research and advancing towards therapies for neuromuscular diseases. So, Nancy, can you hear me okay before I start?
I hear you just perfectly.
I'm just going to turn on the camera for a tiny bit so everyone can see me. So, and I'll turn it on at the end when I answer questions. So for the questions, because my time's limited this morning, if we can just keep it on the topic of what we're doing with the biobank and any questions you have on the biobank, and if you have some specific questions that are slightly off topic, that's okay, but I just ask for them to be specific and not general, because general questions are much harder to answer, or open-ended questions. And just to let you know that the day after the gala event, it hasn't been finalized yet, but there should be an opportunity for patients to ask more general questions and more scientific sort of scientific directions type questions. While today, if we can just keep the discussion to the biobank, I'm very happy to answer any of the questions.
Monkol, let me just interrupt you for 1 second before you get started, and to say, in case anyone doesn't know, NDF is having our gala on November 17th in Los Angeles. Hopefully you've all received invitations to that. And if not, please email me and I'll send you one. I know Los Angeles is a long way for most of you, but we would be happy to have you here. And the day after that, we're having a scientific consortium meeting of the scientists who we're bringing in for the gala. The other thing I would say about what Monkol addressed is if you do have questions that you feel like we're not answering, not obviously Monkol has a specific topic for today's talk, but if there are things that you'd like to see us address in future webinars, please feel free to email either Tara, Amy, or myself, and we will take those into consideration, and with enough of the same question, be happy to schedule a webinar for those topics as well. Our next webinar, just so you know, will be as soon as NIH announces anything about the MANAC trial. We will host a webinar with Dr. Carrillo as soon as we get that information, and we'll get that up to you as soon as we can. Okay, Monkol.
Cool, thanks, Nancy. So the topic today is the genome, Genie Biobank and progress updates, because this idea was conceived just before the UCLA conference last year in 2018. And I just want to update you over the year, what we've collected and how we're progressing with that in terms of, and a lot of people on this call, I've got to thank you all throughout this talk. This would not have been possible without the patients also donating their time and their samples, because a patient collection actually requires patients to actually give stuff. So this is the overview of what I was talking, introduction to biobanks. What is a biobank? Why have one? Why is it actually important? A summary of all the patient sample collections that we've done throughout the year. So from mid-2018 to this point now, where we're at with the blood and skin biopsies that we've collected, and where we are at in terms of the saliva samples, which was reserved for whole genome sequencing performed by PerkinElmer, and then the next steps. And then, and after that, happy to take any questions from there. So onto the introduction and summary. So what is a biobank? So a biobank, for those who aren't familiar with the term, it's just a large collection of biological samples used for research purposes. They're typically collected from people with a particular disease. They're rarely collected from healthy people, because they cost a lot of money to collect and also to maintain. So typically we only collect it from people with disease and their family members. So in the case of gene myopathy and the NDF funded projects, this is collection of blood, saliva, and also skin biopsies, where from there we can actually also process it to have extra stuff, such as the DNA from cells, DNA from the blood, and also DNA from saliva, and also the RNA. So why establish a biobank? It's a great way of centralizing or distributing different cell lines, and these cells are a way for us to understand how gene myopathy works, understand if we have therapies, how they actually work on patient cells and things like that, without actually bothering the patient anymore if we have the cells. So it's a first line of testing without bothering anyone, and also without harming anyone. And it's also ability to actually share it with all the researchers. So to share it with the best minds who have the best ideas of how to study gene, how to understand it better, and also to try different treatments on the cells themselves, again, without bothering the patient. So trying that first, and then seeing in different models how the therapy performs. So this is a summary of the patient sample collections to date. Now, up to date from this morning, because I updated all these numbers. So we've collected blood and skin biopsies from we have 32 samples. So 32 patients and their families have given blood and biopsies at 2 patient conferences, which I'll summarize later on. 8 samples where it was only blood only. Typically, we only get blood only when, if you've been to one of these patient conferences, skin biopsies do take a while to do, and sometimes we run out of time. And also sometimes people get a bit squeamish. It's not the most pleasant thing to actually watch someone get a skin biopsy. So sometimes we only get blood, but most of the times it's because we've run out of time. It's much quicker to draw blood. And we also have 71 saliva samples across 3 different patient conferences also. So I'll show the breakdown of those saliva samples also. So onto blood and skin biopsy. So this is just a little picture of both the procedures where many of the patients on this call have gone through. So in terms of collection, there were 2 major collections, one at the UCLA patient conference last year, so that was around about summer last year, and also the Philadelphia conference earlier this year in May. So in the UCLA conference, the collection was done by Dr. Perry Shea at the UCLA Medical Center, and they collected 15 blood and skin biopsies, and also 7 blood-only samples. While the Philadelphia conference, Dr. Tarsim Mosafa and his team were there, and I think I was there, so I remember they had a small conference room where patients and their families would come and donate. So here we got a lot more blood and skin biopsies because we had learnt from the UCLA experience how to actually better coordinate taking these samples. And so I think this is an amazing sort of like collection in both these patient conferences. So for people that aren't aware of biobanks, to have this much collected in each patient conference is just amazing. And so I have to thank all the patients who and their families who have volunteered to actually give this, to have a number. So my thoughts are, I would like probably 1 or 2 more of these collections for people who considered that the day but decided not to, and also for new patients. So we have a good core set of samples to actually share and understand gene myopathy better. So in terms of the blood, after we collect the blood, one of the purposes of collecting the blood is to extract DNA from it. So we've done DNA extraction on all 40 blood samples that we've collected. So in this biobank, everyone gave blood, and 32 people gave also skin biopsies. So we have DNA extracted. And this is, we have sufficient quantity of the DNA extracted for whole genome sequencing. So a subset of these will go for whole genome sequencing. And this subset is very valuable because not only will we have whole genome sequencing on these samples, so we'll also have cells from these samples and also skin biopsy where we'll be taking RNA. So it's an amazing resource for researchers to understand gene myopathy better. We've also only used a small amount of the blood to actually extract the DNA. So we've kept the rest of the blood in the freezer should other researchers need it for future assays to look at biomarkers that may be important in clinical trials. So this is what happens with the skin biopsy. So that's taking the skin biopsy, it's transported in a flask or a tube, and then we go through this process, which I'm not going to go into detail, to actually extract skin cells from the skin biopsy. And from those skin cells, we immortalize them. So we use some reagents in the lab to actually immortalize them. And what immortalization allows us to do is, if you only extracted skin biopsies and didn't immortalize them, over time they'll all die out because they only have a limited ability to divide and expand before they actually die out. So we immortalize the cells so that, again, we don't have to come back to the patient, ask for more cells, and that we can keep replenishing the cells by letting them divide in the lab before sending it to other researchers so that they can use. So all these cells are stored in the liquid nitrogen. So one of the things we wanted to do with the skin cells is to convert them to muscle cells. And by converting them to muscle cells, we can then study some of the things that are more specific to muscle, because gene myopathy is a muscle disease. So here you can see the conversion. So the green represents a protein only found in muscle called myosin heavy chain. You can see after the conversion, we can see the green. And the green represents just this muscle protein being expressed, and it's normally expressed in this long, thin, sort of like tube, and that represents a muscle cell, which is a lot different to any other cell. And you can see these long thin lines of tube have these blue dots, which represent the nucleus of the cell. So, so this is what we've been able to do, and what we can now do, because we've created skin and muscle cells, is share it with gene myopathy researchers such as Dr. Kelly Harden, who works on gene myopathy. And what she's interested in is using these cells to improve and also develop new biomarker detection assays. And this is important because one of the key questions we're going to ask when we develop therapies is, what impact does it have on sialic acid levels? So by trying it out on patient cells, we have a good way of sort of like developing out these assays and knowing how low the sialic acid levels are in patient cells, and we can also correct them in the lab, correct the patient cells in lab and see what impact it has on sialic acid levels using these various assays. So in this case, it's a lectin assay where lectin selectively binds to sialic acid, and by binding to it, we can measure how much has bound to it as an indirect measurement of how much sialic acid there are in patient cells. So this is a good example, and we've also shared it with Dr. Noah Weisleiter, who's also interested in this case of seeing how susceptible the muscle cells from patients are to damage, as a way of also looking for a biomarker and also looking for a way of, after we've corrected this, the patient cells, what it should look like afterwards, and if it's more like a healthy, normal patient cells. So onto the saliva sample. So again, many people on this call have, or their fellow patients have, given saliva samples, and hopefully I can sort of like sort out the confusion, because sometimes the scientists also working with the NDF are confused. We collected both blood and saliva, and both of them we can actually extract DNA, but the saliva was specifically collected by PerkinElmer for whole genome sequencing. I'll touch upon the questions we want to answer with whole genome sequencing, but first of all, I just want to summarize the 71 samples that were collected and the breakdown. So we got 18 samples from an India patient conference that happened in India, a recent patient conference in Israel where we collected 27 saliva samples, and also the Philadelphia patient conference where we also collected blood and skin biopsy. From that, we also collected 26 saliva samples. So these were marked for whole genome sequencing, and one of the reasons why we want to do whole genome sequencing is that the science advisory board or committee before I joined the NDF were very interested in fully understanding the genetics of gene myopathy. And one way of doing that is to sequence the whole genome of patients, so to get all the genetic variants there are, and ask the question, is there other things contributing to the genetics of gene myopathy? So in a way, we're trying to characterize the genetics of gene myopathy in the context of the whole genome. So what we want to do is sequence all the samples. So we have the capacity and budget to sequence 100 patient samples, and we had agreed with PerkinElmer is how do we pick the best 100 samples to sequence in waves. So the first wave, we were most interested in sequencing all the patients, and also patients where they had multiple family members who were affected, and also patients where we also had both parents. Having both parents sequence gives us a lot extra information in determining which variants appear together by using the parents to actually infer that. So we had preference for that, and that's how 40 of the samples were selected out of 71. It's not to say the remaining 31 won't be used, but we wanted to do it in prioritize what we call waves. So we do 40 samples from there. For the next wave, we want to do 30 or 40 again and pick the best samples we have available, and then another wave with the remaining samples. And the question is to use all the samples that we sequence to actually help us understand the genetics better. And by having this is a great resource that is very attractive to the industry, to have whole genome sequencing, patient cells, and also patient skin biopsies and RNA. So in terms of the next step, and wrapping up the talk before I start with questions, is what are we going to do from here? So with the blood samples, the hope is to use some of the DNA for the next wave of whole genome sequencing. We still have the capacity to sequence 60 more samples. So some of it will come from the blood samples that people have donated. A lot of people have donated both blood samples and saliva samples. So huge thank you for those patients that did donate both. So it's not to sequence them again, but sequence a subset that only had the opportunity to just give blood, that we could actually sequence this for whole genome sequencing. And also, we want to make the blood and DNA available to the gene research community so it can help advance their research by using these samples. With the skin biopsies, one of the things that we want to do before the end of the year is RNA extraction. So your RNA and the sequencing of RNA will allow us to discover other genetic biomarkers that may be too difficult to sense in the blood and also at the protein level. So RNA gives us another sort of molecule to detect for biomarker discovery. The other thing we want to do is further development of biomarker discovery, not our lab ourselves, but by giving these cells to other labs researching gene myopathy or have ideas of additional biomarkers that they can discover. So just to reiterate, the importance of biomarkers are in diseases where it's slowly progressing, such as gene myopathy. Sometimes changes at the molecular level happen before changes at the whole the whole physiological level happens. So in other words, before you can notice improvement in your muscle, other things will actually change on a very small level. And these are much harder to detect, but they actually happen much earlier. So it's being able to know if we're successful with a treatment earlier rather than later. And this creates sort of like encouraging results if we can detect that. So what people have used in the past is sialic acid levels. And by detecting other genetic biomarkers, we can actually have sort of like a range of things that we can check. And lastly, we also want to make this available to the gene research community, all these resources. Last is the genome sequencing and saliva data. So we only just completed the first round of sequencing. The data was made available last week, so I'm proud to say that. It just happened to be in time for this webinar. So both Perk and Elmer and my group will be doing preliminary analysis over October and November, and hope to present some of the results in the November gala, or particularly the day after the gala, of how the results look like. And from that, we can better plan the next wave of sequencing from what we've learned from that. And again, also to make the data available to the gene research community. So each one of these are valuable resources in terms of the cells, the DNA, and also the data. And I think by making it available to the community in an open way, that it will help advance gene research. One thing I did not mention throughout the talk, but I do want to mention right now, because I think this is a very important point to make to the patients, particularly on the call that have generously donated, that at all times these samples remain de-identified to the researchers, and that way people's privacy is protected. So all these samples are labeled with just letters and random numbers, or sequential numbers, albeit sequential numbers. No one knows which patient actually belongs to, and this is the way we protect patient privacy. So with that, I'd like to thank everyone, particularly the NDF team and donors, for making this research, this important research possible and available. Also patients and the families, because without the patients and family, we wouldn't be able to actually build up this biobank. And lastly, the gene research community for their support and also for the future use of this resource so to advance their research and also therefore advance our knowledge in gene myopathy. Thanks.
Thank you, Monkol. And I think just so you all know, we have a couple of questions. Feel free to type them in if you want to, and I can also unmute anyone who wants to say something. But with regard to the gala and the scientific consortium meeting, Monkol has been a large part of our ability to create a collaboration among scientists all over the world who are now working together in ways that never happened before, in part because of our ability to create these samples and for our ability to have data that we are willing to share with them if they're willing to share theirs. So I think, Monkol, you would agree that scientists are working together now in a way that we haven't necessarily seen before. And at our gala and at our consortium meeting right now, scheduled to be there, Dr. Nishino, Dr. Rosenbaum, Dr. Argov, Dr. Carrillo, Dr. Hoising, both Dr. Lex, Doctors Lek, Kelly Harden, who Monkol spoke about, will be there, Dr. Mozafar, John Hakimi, Dr. Weissleiter, and hopefully some representatives from biotech companies. So if you are able to be here, obviously it's going to be quite a gathering of important people, and we hope that the consortium meeting will have the kind of results that our meetings have in the past when we get these scientists together. Some real important decisions and conversations are had, and I think that the fact that everyone's talking to each other with us as sort of the hub has led to great progress. We have a couple of questions, Monkol. Maya is asking if you need more saliva samples, especially from big families.
Yeah, I think that's a fantastic question from Maya. I think the answer is yes. So we can either, one of the things on my to-do list on the next wave of samples is to reach out to Nishino in Japan, and also to reach out to Stella and Dr. Argoff to see if they actually have them. Otherwise, another way we could actually get them is to reach out to the certified patient advocates such as Maya to actually get that, because with larger families, we actually have more variability and also power to detect sort of like subtle changes that could help with the design of clinical trials, because I think Oksana has presented before in a few different conferences that I've been in, the relationship between your mutation and how severe the disease is and its progression. So the answer to that, Maya, is yes, it would be very helpful for the next wave. The other thing that has been helpful that I haven't touched upon is another set of samples we are going to sequence is the efforts by Sibash and his collaborator in Iran. I can't remember his name off the top of my head right now. Nancy may know. Who have collected some really, really interesting families with variable gene severity. And so we'll also have them sequenced because they are very valuable samples. Amongst those samples, and Maya, if you know of other families, it would be great if you get in contact with India. We're interested in some families where they should have gene myopathy based on their mutation, but they don't. And they're quite advanced in age, so they're in their 70s and 80s. So our collaborator in Iran, through Sabash, had actually collected some of those samples, and by understanding why they either don't have gene or myopathy or why they're getting so late can help us understand other avenues of research or other pathways we can target to treat gene myopathy. So if there's anyone on the call that does know those of those families, do get in contact with the NDF because they are a very, very interesting subset. So Maya, you or some of your fellow patients.
Yeah, I think you're talking about Hossein Kademian, right?
Yes, that's it.
Yeah.
So he did collect some interesting samples, and we will, aim to sequence it. Maya, that is also a great question. So um, I'm hoping that PerkinElmer can actually send it out to the families if we do identify them. And um, it's not just a matter of just sending out the kit. They also have to consent. So they'll probably just send out the consenting paperwork. And, if need be, myself or someone else can help with the consenting process. So it's just to make sure that patients understand um, what will happen to their DNA and that their privacy will be detected. So the Saliva kit shouldn't be too much of an issue. And they're also commercially available to buy off the internet. It's more the consenting process itself.
Yeah, we can definitely coordinate that when we identify people who we need them sent to. We were able to send some home with people for their family members when we were in India, who then sent it in to PerkinElmer, and PerkinElmer has labs in different places. So we
So
should, I think we'd be
I
able
think
to.
that answers Roberta's question about sending a saliva kit. So I think for interesting families, we definitely, could arrange for a saliva kit to be sent to them. And that is the great thing about saliva kits. They can be sent in the mail, and the saliva does get preserved at room temperature when it gets sent back. So I'm familiar because I sent a saliva kit. I got a saliva kit sent to me, and I sent it back. That's how 23andMe does DNA extraction.
Right. Are you able to see Michaela's question? Because those words are too long for me to be able
Yep.
to.
So yeah, so that is a very specific question. So I'm not the best person to answer the questions regarding lectins, but I can go back to these slides, and you can see the different lectins from Michaela's question. So the thought is that lectins bind to different combinations of sialic acid. So Kelly and also Marianne are much better than me answering this question. So I'll try my best to answer it. That the thought in this table here that from Kelly's sort of like research is that different lectins have a different affinity for different types of sialic acid or different versions of sialic acids bound to other things. And the thought is to give each one of those a try, that some of them may be better at the specific question we have. The specific question is, can we tell the difference between a patient with a particular gene mutation and healthy patients? So some of them can all of these definitely can bind sialic acid, but some of them can't tell the difference because sort of like their sensitivity is not good enough. And I think that's the main thing that we're most interested in terms of lectin and its binding to sialic acid. So to go into the science a little bit more, I'm probably not the best person to go into that, but that's just trying to explain what we're trying to do with the different lectins.
Yeah, and we can certainly have Kelly come on for a webinar sometime to explain specifically the work she's doing if people would find that helpful.
Yeah.
I know she'd be happy to do that. Tara was asking if there will be any kind of report specifically to patients about their own samples.
That is a great question. I think I'm not 100% sure on the agreement with PerkinElmer, but PerkinElmer, from what I remember, but Lali would be the best to answer this, was going to produce a report, but I'm not sure the report was a scientific report or to go to the patients. But one thing I do want to clarify to everyone on the call and also Tara is that um, what we're doing in terms of the science behind the genome sequencing, we're not doing it to actually get a genetic diagnosis for patients. So the people we're recruiting already have sort of like a genetic report to say they have gene myopathy. It's more to understand the other genetic variants that they're actually carrying, as I think Roberta has actually touched upon in her question.
Yeah.
Um, So, so Roberta, unfortunately, due to patient privacy, I don't know who the samples Hussein has collected, who actually it's from, and to comment on that because of patient privacy.
Yeah, Can you hear me, Monkol?
Yep.
Yeah, hi.
Yep.
I guess last year he came to Turkey and we met.
Yep.
He wanted to collect my blood samples from my mother and from my father.
Yep.
And he wanted to just look for that for himself. And then we talked about results. I have only the chat, our chat
Yep.
mail, sorry.
Yep.
And then he told me that, as far as I can understand, I have two more mutations that has two more muscle diseases. I guess it was myofibrillar myopathy, and I don't remember the other one. And he told me that we should look that if they are active or not. And if you are talking, working together, can you take my samples or take his works? I don't know. I want to participate to this research. That's why I'm talking about this.
Yep. I think it's probably best to coordinate not well, how about you drop me an email afterwards and we can discuss further.
Okay, okay.
Thank you, Roberta.
No problem.
Does anyone else have any other questions for Monkol while we've got him here?
I'm just scrolling to see if I've touched upon.
I think you're good. I think you have. I think that your presentation was so thorough that no one is left with any questions.
I think Michaela has a question.
Monkol, I have a question there that you haven't seen. This is Tara.
Yep.
So I was wondering, with the samples you have tested so far, are you seeing any significantly statistical pattern
Oh, so
in terms of mutations, in terms of novel mutations, in terms of any
yep.
gene-gene interaction?
Yeah, that's a great question, Tara. And sorry, yeah, I did see it, then I totally forgot that I saw it. So that's a great question, Tara. So at the moment, the sequencing was only completed a few days ago, and that's what we do want to look for. One of the challenges, though, and I've always presented this in scientific meetings on gene hemopathy and also in patient conferences, my worry is maybe 100 samples may not be the word for it is well powered to actually find statistical significant findings. We may find findings that are at the sort of like at the threshold statistical significance, but we're definitely going to look for that. Like, are there genetic variants that go with gene myopathy, pathogenic variants that may help us understand the disease better? But as I said, the sequencing was only complete a few days ago, and the data was only transferred a few days ago. So PerkinElmer will be looking at it and using their range of established analysis, and we'll probably look at it here at Yale also because it's one of my research interests. But we'll keep you updated on that.
Well, thank you. And the follow-up question on the saliva kits, are you testing family members as well, or just the patients?
Good question. So, so maybe I didn't explain it that well. So this is a good opportunity to explain it again, Tara. So at the moment, for the first wave of samples, we just did affected patients, and the only extra samples we did was if we had the parents also, both parents, and also we would do, if there were multiple affecteds in the family, we'd do all the multiple affecteds. The reason why at the moment the only unaffected we've done is parents is because it allows us to do, there's this term in genetics called phasing. So it allows us to know which chunk of DNA came from mum and which one came from dad. Without the parents, it's hard to actually do that, this term called phasing. While if you did an unaffected sibling or unaffected, family member, other family member, you can't actually do that. So the unaffecteds we've done so far are the parents. The other reason why is we only have 100 samples at the moment contracted to do. So I think the best genetic study design is to do the affecteds. It's not to say that we're not going to use the unaffected. The unaffected will be used once we have an idea, like your other question, Tara, once we have an idea of particular genetic variants or genes that may be contributing, we can then use the unaffected to test this out and get better statistical significance, but in a targeted way. So we may not do whole genome sequencing, we may just do sequencing of a particular gene.
So just to clarify, both parents have to be alive for you to take the samples from the affected patient.
Yeah, so it's much harder with GD myopathy patients and also other neuromuscular diseases that are later onset, because if you're 40 or 50 years old, that means your parents are older than you. So sometimes it does get tricky, but definitely affected patients who have donated saliva have been sequenced in the first wave. So out of the 71, we did all the affected patients, provided the DNA was good enough. So it was only one sample where the DNA wasn't good enough. Sorry, I have allergies today. So Michaela has a question then, but I'll answer May's question because it's a very quick one. So that is a good question. So it's up. I'll have to talk to the NDF because I think it'd be a good opportunity if there are some people that haven't donated saliva to do it at the gala. If we could get some saliva kits there.
Yeah, I mean, I think we can see who's coming and see if we think that there's samples that we don't have yet. I don't see why we couldn't arrange for that, or even maybe, I don't know, I have to look into this, but maybe having patients go to Perry Shea at UCLA while they're here.
Oh yeah, that'd be a
saliva.
fantastic idea too. So, but just to clarify to everyone, we don't need you to donate twice. You've been generous already with the donation. So we only probably need 1 or 2 patients to donate twice when we weren't able to get good DNA from the saliva.
So I think with regard to the gala, we would take it on a case-by-case basis, seeing who's coming and how they fill gaps that we have, and you know, their time and willingness. Obviously, saliva is easy enough. I'm sure we could get kits sent to us and take care of it ourselves right then and there. If we want to talk to Dr. Shea, you know, we can consider that too, depending on specifically who, you know, which patients we're talking about.
Cool. The only thing I'd like to say, if you're you know, if you're giving saliva at the gala, do it before you eat.
Yes. Well, we'll see who's coming, and we'll talk specifically to those people offline and make sure we make the proper arrangements. And you see a question about collaborations.
Yeah, so I'll try and answer, Michaela. So some of the new collaborators we're brought on board is Kelly Harden and also her former mentor, I've just gone blank. Paul Martin. And so I think they're a fantastic addition. So Angela Lek, also my wife, and working with as the co-director, the co-scientific director. When she visited OSU at the end of last year, she met Kelly and realized that Kelly was working on gene myopathy and also Paul Martin. We had never known, and it's been fantastic to bring him on board because Kelly, like Stella, had actually worked on AAV and gene therapy on the gene myopathy mice. And so just to let you know, Michaela and others on, it's great to also have not just Kelly, but Paul Martin involved, not as direct as Kelly, but involved also. By having them on board, it allows us to push forward with some of the gene therapy efforts, which isn't the sort of the focus of this, and we can talk more about it at the day after the gala event where Kelly's there also. So because they bring in that expertise, because they've worked on the mouse like Stella. And the other thing Paul Martin brings is he's also creating a mouse model too, along with Stella. So it's a way of sort of like hedging our bets, because for us, it doesn't matter who creates the mouse first. A new mouse first for us to study gene myopathy, to study gene therapies, and sort of like gene myopathy. The thought is whoever creates it first and it's working, to share it with to share it with all the researchers around the world, so we can get to where we need to be quicker. So hopefully that answers your question, Michaela.
And we've also had recent meetings in India with the Bhattacharyas and everyone in their organization. And also Lali went to Japan and met with Dr. Nishino, and there's all kinds of conversations in the works about collaborations with them as well.
So just to add to a comment Nancy said just before I started talking is I'm very excited. I joined at the NDF around about the end of first quarter of 2018, just to see the amazing sort of like collaborations that the NDF have done in that short amount of time, that year and a half. It's just been amazing to get all the players, all the best researchers around the world all on the same page and talking to each other. It's just been amazing effort.
I agree. We've been busy, and we've been busy on all kinds of fronts, and with lots of progress coming our way, obviously. And with Monkol's leadership in the scientific community, it's really been an essential part of what we've been doing. And we hope that with the money we hope to raise at the gala, obviously that's another important part of what we're doing. We can match that $2.5 million matching grant that we received last year and continue to fulfill the grant requests that are coming into us. We can make this the last question, Monkol. Michaela's asking about.
Yeah, so I'll answer Michaela's question. I'll just add to what you just said, Nancy. You know, the generous donations is one of the driving forces of bringing the community together, because everyone has great ideas and research projects that at the time couldn't be supported by other means, and it's another way of bringing the researchers together, and also not just bringing them together because NDF has generously supported their research to actually push them towards more collaborative, what I call open science. Open science is when you have a result to share it with everyone. By sharing your results early and with everyone, we can get to where we need to be quicker rather than just holding it to yourself. So I'm going to answer Michaela's question. So this is another great question, Michaela, regarding metabolomics. So unfortunately, the blood that we've collected, I discussed this with some people in Maduri's lab back in Emory, and I said, when we collect the blood, can you tell me how you want us to store it? And we haven't had we haven't been on the same page at that. So it's the hope that future blood collections that we can actually store it in the best way so we can do as many experiments on them. But us keeping it frozen at negative 80°C, we can still do more experiments on them, but not the biggest range of metabolomics assays. So it's the hope that the future collections we can start doing that. But it's a great question. It's something we're thinking about for the next blood collection, because again, the hope is to only bother patients once.
Right. All right. Thank you. And I wanted to just piggyback on what Monkol said about the scientific consortium and, the funds that we've been able to raise and how that has positioned us so well with the scientists that we're working with. Part of the agreement with this open science idea is that in order to be a grant recipient and in order to receive data from us or from any of our other scientists that we're working with, you have to, agree to have your scientific studies shared as well. So everyone is sharing with everyone. And I think it's created a collaborative environment among the scientists that's really helping to spur our progress. And Monkol, I think you would agree with that.
Most definitely. There's just thank you everyone for your time. I just want to get one more thing in. I'm sorry everyone. And that's for me to also say how impressed I was with the efforts of Tara and also Amy in actually creating a patient driven led registry. The registry is amazing. So I've given Tara and Amy some feedback of extra things that we could improve upon it. So just to let everyone know on the call that, thank you for your time in, contributing information. And, we're almost there. And there's certain things that I haven't had the time to work closely with Tara and Amy, but hope hope in the next few weeks so we can, get it to exactly where we need to be, because this registry is so important in terms of when we approach industry to say we have this many patients around the world and this is sort of like how they're located. But again, always protecting patient privacy and things like that. It's something that a lot of diseases do not have, and that's why I'm impressed with the hard work of Tara and Amy and also the NDF in actually creating this.
You were a mind reader, Monkol, because that was going to be my last point. I'm glad that you brought it up. For any of you who haven't participated in our patient registry, it would really be great if you could. Obviously, Monkol has explained why we need the information. There's a link on our website, and you can fill it out online, or you can download the form. If you need it translated into a different language, we're happy to do that as well. Just contact Tara or Amy, or send an email to info@CureHIBM, and we'd be happy to help you facilitate getting included in the registry. Super important part of what we're doing. So with that, I want to thank Monkol for taking time out of his Saturday, and thank all of you for joining us today for what I hope you found to be an informative webinar. As I mentioned before, if you have topics you'd like to see us cover in addition to the huddles that we host monthly for patients specifically, we try to do these webinars a few times a year with specific guest speakers, all in an interest to educate and enlighten our patient community. So please feel free to keep your suggestions coming to us and let us know what NDF can continue to do for you. First and foremost, obviously, we're a patient advocacy organization, and you're the people who we're working so hard for on everything we're doing. So I thank you for joining us. We've recorded this session. It will be available soon, and we'll let you know when that happens. Please stay in touch, and if at all possible, we'd love to see you here in November. Thank you, everyone.
Cool. Have a great day, everyone.